But you can move it if you want. Like,
Yeah,
sure. Yeah, feel free. And then you mentioned the chat, right? You mentioned the chat in your little intro there about people are muted, but if they don't want to forget their questions.
Yeah.
What time we got? 11:55. 11:55. Exactly.
Pink. Please go on. We will start in 2 minutes. Okay, good afternoon, ladies and gentlemen. It is time to start. Welcome to the Office of Information Management and Analysis Brown Bag Series. I would like to remind you that OIMA Brown Bag will be held every month. This is the second in the series. It will be strictly a 30-minute event for both presentation and question and answer. Today's seminar is about contaminants of emerging concern. The speaker is Dr. Keith Maria. Keith is the principal scientist for chemistry at SWRCB, the public research agency that focuses on water quality, investigates the measurements, fate, and effects of environmental chemicals, including contaminants of emerging concern in ambient and recycled water. Keith is a leader in developing and applying bioanalytical screening tools, non-targeted analysis, and passive sampling methods for water quality monitoring and assessment. A native Californian, he was a research professor at Skydaway Institute of Oceanography in Savannah, Georgia, prior to joining SWRCB in 2005. So with this brief introduction, I'll pass the phone line to Keith to start his presentation. Thank you, Keith, for doing this for us.
Well, thank you, DeWitt, and also Eric and the rest of the folks at OIMA. For those of you who are familiar with the work we've done, especially down in the statewide and in the Southern California regions, hopefully some of this material is not the first time you've seen it. The conundrum we're faced with, what we call contaminants of emerging concern, is that there's really potentially a very, very large number of them. As you can see in this graphic, the number of chemicals that we know about that are sort of registered in commercial and consumer uses, numbers in the several tens of thousands, estimates of 100,000, 150,000 chemicals out there, really puts us in a challenge to sort through the chemicals that we really should be concerned about and that we want to incorporate in our monitoring programs. But with that very large number, what we actually monitor for both in the ambient environment, an example is our own Southern California Bite Survey, we measure probably a couple of hundred individual chemicals. And in drinking water, we measure probably double that amount. We have the capability to measure these things with current analytical techniques, but the development and validation of these techniques, as you guys are well aware of, takes a while, takes an investment in time and money. And with the tens of thousands of potentially interesting chemicals out there, this chemical by chemical assessment paradigm becomes a bit of a weight, an anchor around our ankles to keep up with being vigilant with chemical monitoring. The other thing that we're faced with is these chemicals never occur individually alone in the environment. We always see mixtures, and the mixtures can vary depending on the source, whether it's treated wastewater effluent, whether it's stormwater runoff, whether it's leaky septic systems. So we've got a kind of a chemical soup out there, and that soup can vary, again, in space and time. The other thing that's challenging is that we're learning more and more that some of these chemicals can have unwanted effects at pretty low concentrations, and you'll see some of the numbers later on in the presentation. We're talking about toxicity information in the parts per trillion type range. Now, I don't want to be a doomsayer because, again, our ambient monitoring to date shows that, hey, there's no immediate damage that we can see with our current techniques in monitoring, both in situ and in the lab. But the thing that becomes challenging, though, is looking for these low-level chronic type effects. So that's another challenge we're faced with. Liken it to, again, this chemical soup of how do we deal with chemical mixtures. The other thing that we're learning more in as we start to reclaim more and more water in the coming decades, this is going to become a very important undertaking in California. We start to realize that we cannot remove everything, and even if we can remove everything, our processes for treating sometimes create transformation products that are hopefully less toxic, but they may have the same or even increased bioactivity. And we need just to understand that. We have examples from the past where, for other purposes, we're making the water safe by chlorinating it for microbes, but the unintended consequences of that is we may form transformation chemicals or new products that we really, again, should understand and keep a handle on what their potential effects are. Again, this is going to become more and more important as we recycle more water. The treatment processes involved with recycled water are very energy intensive. They're very good at what they do, but there will be some issues such as brine discharge, maintenance of treatment processes like RO that require some disinfection, some chloramine addition. And again, we just need to have some good tools to get a handle on what that means for eco and human health safety. So, it's already been 6 years since we started the CEC statewide effort that SWRCB has been involved with the State Water Board and the swamp folks. So back in 2010, we really didn't know even where to start, and so we convened a panel of 6 experts, actually 7 experts, on behalf of the State Water Board to sort of guide us and give us recommendations on what we should be looking at and where are the gaps in knowledge that we should invest our money into. Because the data at that time was pretty scarce, the list of CECs that could be recommended for monitoring was pretty small. About 16 individual chemicals were prioritized for measurement, individual measurement. The larger recommendation, though, was what are we going to do about new chemicals, the unknowns, the transformation products? Can we afford to develop individual measurement techniques for these things? Do we even know what they are? The answer was pretty much no. And so the bigger recommendation that came out of the expert panel was to develop some more comprehensive screening tools, and we'll be talking about these in the next few minutes. The other thing that was very apparent is the activity of some of the chemicals of concern. We have hormones, both natural and synthetic, that we know can have affect wildlife at pretty low concentrations. Again, we're talking in the parts per trillion range here, as you see in the 17-beta estradiol. The NDMA that you see in red is actually a byproduct of water treatment, and it is a carcinogenic chemical of concern, again, at fairly low levels. And so our techniques for screening these chemicals need to be pretty sensitive. So where did we turn to first? We noted that we're not the only ones interested in measuring the activity of these CECs at pretty low levels. Government research, in particular EPA, has been developing what we call bioanalytical screening tools or cellular assays. These use engineered cell lines. Many of them are human-based cells that will respond to the exposure of an individual chemical. More importantly, they don't discriminate between individual chemicals. They sense chemicals based on a common, what we call mode of action. So, for example, all environmental estrogens can be screened using a certain number of these cell lines that will respond to estrogenic responses. And what it does, it integrates both the exposure, let's say the transfer of that CEC on the left into the cell. So we know it gives us a measure of exposure, But then it takes us to the next level and says, what does that exposure do? It initiates some kind of cellular response that takes us along to what we want to know about. And what we want to know about is this going to cause some effect that we're concerned about, whether it's a reproductive insult, whether it's cell death, whether it's some other higher order effect. These cellular assays are actually in the red there, the initiation step to what we call these adverse outcome pathways. So it's the start of a cascade of events that gets us to an effect of which we're concerned about. Previous technology, you look up in the upper right of this slide, you see that chemical binding. We've had assays for decades that will tell us that a chemical will bind to DNA or a certain protein, but it did not tell us what its role was in these adverse outcome pathways. And that's the real additional relevance that we're seeing in these bioanalytical screening tools. So what's available right now, what have we been working on in the last 5 years? There are a fair number of these bioanalytical assays that have been developed. We do not develop them. There's a whole host of cellular biologists that have engineered multiple cell lines to screen for different classes of chemicals. And again, they do this by mode of action. They don't care whether it's 17 beta or 17 alpha estradiol. They'll screen it by class or mode of action. And here are some of the more important ones that are available right now. You see in the yellow are sort of these endocrine disrupting type cellular screens. We've got cell lines that will screen for genotoxicity and also aromatic hydrocarbons that can lead to things like genotoxicity and eventually cancer. So we've got a handful, dozens of these cell lines that we have been trying to adapt for water quality monitoring. And some of these are showing great promise, and that's what this slide is showing. We took a commercially available cell line that will screen for estrogenic compounds, and what we did was we tested it with a range of different water qualities. We took secondary effluent on the left, and we showed a fair amount of, we can detect the estrogenicity in that, as you can see on the far left. We treated that effluent with membrane filtration, and we were able to reduce that activity, but again, it's still detectable. But when we get down to finished waters and even ambient waters, we see that the estrogenic activity drops pretty much below detection. And this is what we see if we do individual chemical measurement as well. So what we're showing here is that these screening tools give us the expected response for different water qualities. The second thing we saw and we wanted to show is to determine its tech transfer potential. These cell assays are no good if only one research lab in the world can run them and nobody else could. So we put together a team of different labs to see what kind of inter-lab agreement we get. And by and large, we were able to get pretty good across-lab agreement with these cell assays. The next challenge we have is that, okay, if targeted chemistry tells us exactly what the bioassays tell us, you know, what good are they, what kind of added value do they give? This plot here shows along the y-axis, if we measure individual estrogens and add them up, add their activity up, do we get the same expected response that the cell bioassay that's plotted on the x-axis gives us? And for the estrogenicity assay, we know many of the chemicals that will respond in that bioassay, so our correlation can be pretty good. In other words, if we measure 12 estrogens, 12 of the more potent or occurring estrogens, we should be able to track what the bioassay tells us and vice versa. But that's not true of some of the other mode of action assays that we're looking at. The glucocorticoids are a class of steroids that we use in topical creams for histamine type responses, so they're reduce inflammation in tissues. We know that there's quite a bit of activity for GR in wastewater effluent. As you see on the first column, the mean BEQs are a measure of that response. But we don't know the chemicals, the individual chemicals that cause that response, and that's in the middle column. You see, we can only really explain less than 1% of the activity if we measure the active chemicals in the sample. So we need another tool to tell us what bioactive chemicals are responsible for some of these screening responses. And that's where we're working on something called non-targeted analysis. The monitoring we do now targets individual compounds, whether it's a PCB or arsenic or mercury. We know what we're looking for. We can fine-tune the method to give us a robust, very sensitive measurement of that. But when we do that whole process, and not just on the instrumental side, but when we preserve and process or extract the sample, we discriminate for mercury or arsenic or PCBs. In other words, we eliminate everything else is in the sample. That's represented in the yellow bars, or yellow fraction of bars. So we miss a lot of stuff that's in the chemical mixture. Non-targeted analysis simply tries to recover and retain the sum total of chemicals that's in a given sample, and we use high-resolution mass spectrometry to identify these individual chemicals that we typically don't look for. It is a very powerful technique, but as with any new technology, it does have its disadvantages. Equipment is expensive. It generates oodles of data that need to be processed and interpreted. So in that sense, currently, you need people that are doing this pretty much on a daily basis to create libraries of these new chemicals. So we're starting to do that with non-targeted. One of our first forays using non-targeted was, again, not in water per se. We were interested in what kinds of chemicals persist and bioaccumulate. So in the marine environment, we went to the top predators. We're working with our collaborators at the National Marine Fisheries Service in La Jolla. We were able to get a bottlenose dolphin samples, blubber, from animals that frequent the Southern California coast. And the basic idea for selecting these dolphins is they're kind of like human sentinels in the ocean. They're long-lived. They're high on the food web, so they eat prey such as fish, and basically they have the potential to accumulate fairly high levels of persistent contaminants. What we found when we applied non-targeted analysis to these dolphin samples was we do see a lot of chemicals that we don't monitor for. Again, you remember the list for the bite was 200. About 100 of those are probably organic anthropogenic compounds like PCBs and DDTs. The light gray bar here shows that there are equal number or greater number of synthetic organic chemicals that we don't routinely monitor for that exist in these sentinel animals. Moreover, we see a number of compounds that we don't even know what their structures are yet. So we have to continue investigating what these things are. And again, the key thing is, are they relevant at all? We really don't know that answer. How do we talked about two tools. We talked about these bioanalytical screening tools. We talked about non-targeted analysis to help identify bioactive chemicals. How do they fit into current monitoring? The light blue boxes are basically the existing monitoring paradigm where we do targeted chemistry on samples as an exposure screen. If they exceed a guideline or criteria, then that requires some additional action. Usually that action involves whole animal testing. And in many cases, whole animal testing will tell us, hey, no big deal, we don't see any effects, therefore we can conclude that that exposure is not resulting in something that we actually care about. We combine that at the confirmatory level or tier 3 level in this picture with what's going on in the ambient world. You know, we do our in situ surveys, we look at trawls, we look at fish populations, we look at the health of invertebrate communities to make sure that they're at some level that compares with a background condition, whether it's a 20% degradation or whatever the policy number is. We want to make sure that in the real world we're not seeing populations crashing or damage that would cause concern. But the two tools that we talked about, we think that they will both enhance and make monitoring more efficient and comprehensive. The targeted chemical analysis, again, is limited to the chemicals that we have methods for. So if we incorporate these bioanalytical screening tools at that screening level, the first level, we can not only screen for a greater number of CECs and chemicals, but that information will also inform what types of targeted chemistry we should do. If the estrogenicity screen lights up, we can immediately start looking for estrogenic, known estrogenic chemicals. Okay? So it's a double benefit in our minds of using and developing these tools at the screening level only. Will they tell us right off whether an animal will have adverse effects? No, definitely not. So there's still a role for higher organism toxicity evaluations. We're not suggesting that these things will replace that, but it should help inform that and hopefully in the future reduce the number of whole animal toxicity testing that we need to do. For the non-targeted chemistry, we see it as a very powerful diagnostic tool. When we do see hits in our screening level and the targeted analysis does not explain that, we can broaden our search, broaden our scope, and look for, focus in on those chemicals that are causing the bioactivity or the toxic response at the whole animal level. So this is how we see enhancing our monitoring with these two tools. So currently, we, well, about 2 years ago, working with DeWitt and Rich and the folks at Swamp, we took these recommendations and we scoped out how to test drive these tools or this enhanced monitoring framework in various parts of the state. So we put together this planning document, talks about the targeted CECs that we should be measuring moving forward. It talks about how we can evaluate these bioanalytical screening tools and the non-targeted analysis in different scenarios around the state, whether it's an inland water, whether it's a coastal embayment, or whether it's offshore marine wastewater discharge. Some of the case studies that we're now working on, actually we learned a lot from San Francisco Bay Region 2. They've been looking at CECs since the 90s, early 90s. And so we can go to school on what they've already learned in designing our current pilot studies. At the same time, in the Bight, in our neck of the woods, some of the first endocrine disruption type research or surveys were done for the third Bight exercise, which started in 2003. Again, we can go to school and learn from those early pilot CEC projects. What we're currently working on, though, are two studies that just started late last year, one in Region 1 on the Russian River, and the other as part of the perennial stream assessment, but in Southern California specifically for the Stormwater Monitoring Coalition study. So just some very preliminary results from some of these pilot studies. In the LA region, we actually started working with Mike Lyons and Deb Smith back in 2011 to get a head start on the targeted side of CEC pilot studies, taking the list of priority chemicals that the panel recommended and looking for these compounds in the effluent-dominated systems that we have. We've done work on the Los Angeles River, we've done work on the San Gabriel River, and this slide shows our latest results from the Santa Clara River, which differs a little bit from the former two in that it's got a lot more semi-natural habitat. It's got a lot more soft bottom and natural riparian habitat along its length. Basically, we want to see what happens to discharged CECs from the wastewater treatment plant to the water reclamation plants, and whether they persist in stream or whether they're attenuated rapidly or not so rapidly. In the previous studies, the channelized systems in LA County or at the LA and the San Gabriel rivers, they're pretty much open pipelines. And there's not a whole lot of opportunity for degradation. We saw a little bit different story in the Santa Clara River where downstream from the two wastewater treatment plants, the WRP number 1 and WRP number 2, we saw pretty rapid decline in the water concentrations. When we look at sediment, though, the sediment story is a little different. Depending on the chemical, the red would be trichlocarbon. We see that chemical wanting to be more in the sediments downstream of the wastewater treatment plant. So we do have to consider the chemicals individually in their behavior, whether they like to be in sediment or whether they're subject to photo degradation and whatnot when we want to predict what happens to them downstream of wastewater treatment. I mentioned the SMC study. We are now applying the bioanalytical screening tools to inland waters collected as part of that perennial stream assessment. These are results from about 30 samples from inland watersheds in Southern California, and these are results from the same estrogenicity or estrogen receptor test that I showed previously. The good news is a lot of blue and green, so our responses in these waters themselves are pretty low. In the couple of samples where we do see something at the 5 nanogram per liter level, which may be a threshold where you want to investigate a little bit more, we can use non-targeted to see what's different in those samples compared to what's in the samples that gave us no response. So that work is underway. In Region 1, working with Rich Fadness and the folks up there, they have been monitoring our panel work and the pilot study guidance exercise. So they jumped on board and say, okay, let's answer some of these questions about CECs. Are they in our system? And then let's help you evaluate your new technology. So we're appreciative of Rich and the region up there for stepping up to help us pilot these things. This study really just kicked off. We did collect fish tissues through the Moss Landing Marine Lab. We analyzed for flame retardants, the polybrominated diphenyl ethers, in our own lab here at SWRCB, and we don't have the data to share, but I have seen the data. They are pretty low concentrations, in the low ppb from what we have now. It's similar to what we see for the DDTs and other legacy organochlorines in those fish. So there doesn't seem to be anything really hot from what we've seen so far. Our future plans, any day now, if it ever rains again, I thought this was El Nino year, but we are going to apply our bioanalytical screens and non-target analysis to stormwater samples and wastewater treatment effluent from that watershed. again, the messages are we want to be vigilant for CDCs. We don't see a need for knee-jerk reactions. We're not seeing our wildlife crash. There are no smoking guns out there. But to be vigilant, we definitely think that some of these screening tools are the way to go. We still need to expand the number of cell assays and mode of action that we can screen for. We only have a handful. Maybe we need a dozen, maybe a couple of dozen. But as we develop technology, that screening will become more efficient. We want to develop the methods and make non-target analysis, again, more efficient. I alluded to it requiring a lot of data analysis, and we're hoping to automate and get groups together that work on non-targeted to put their heads together and come up with common catalogs and databases so we can have a reference library, so to speak. And then we want to test that framework to see if these screens will actually inform our current monitoring approach and whether these non-targeted methods actually can identify substances, whether they're in ambient waters or as a part of water treatment, help identify which processes and which contaminants should be added to future lists. With that, I think I'll stop. I want to, again, thank DeWitt and Eric and the OEMA group for sponsoring the pilot study planning work. Some of the regional folks, Mike Lyons, Deb Smith, and Rich, I mentioned. Also the people that are...
All guests have been unmuted. All guests have been muted. All guests have been unmuted.
And, yeah, Alvina is there with the, she's our scientist with the tools and big audit.
Okay, thank you, Keith. Unfortunately, we don't have much time for questions. We want to keep it strictly 30 minutes, but I'll give like 3 minutes time for people to ask if they have any burning question right now.
I'm sorry for going over. I didn't realize I went over. I'm sorry about that.
Yeah, that's okay. Anybody has any big question for Keith? Okay, thank you very much all. Next time we'll be on time. The next webinar will be March 30th. It will be on Wednesday. And the announcement will be out soon. Thank you all. Bye.
All right, thanks.
Left the meeting. The meeting is